Hey guys, today we'll be going over the second week of class, which is basically the cell membrane. And the cell, like we know, is the smallest unit or structural component that has the ability to carry out all the processes of life. So each activity on the cell depends on uh, specific structural properties of the cell. And if we're looking at organisms like us humans, which we are multicellular, we understand that cells are the living blocks of those organisms. So an organism's structure and function will ultimately depend on the individual and collective structural characteristics of and functional capacities of our cells. Part of the cell theory is that cells are going to come from other cells and they will give rise to other types of cells. So cells all come from pre-existing cells until we go far enough to the first living organisms, which arose in a situation that we're not really sure of, but they did happen sometime and therefore past. So cells of all organisms are fundamentally similar in structure and function. So here we have a typical eukaryotic animal cell with the different parts. I will tell you as a recommendation to go over each organelle and say at least their main function so we can understand this and go on. But we're going to spend more time specifically understanding that eukaryotic cells all have a cell membrane that separates that between the ECF, extracellular fluid, and intracellular fluid. We have a cytoplasm, which is what's inside the cell, this colloid structure that permits the movement and structure of the cell. And hidden away in a double lipid bilayer, double membrane, we have the nucleus, which houses the DNA of that cell. So plasma membrane, also called cell membrane, surrounds every cell, creates two different compartments, what's inside the cell, ECF, and what's outside the cell. And this will be a structure that is selectively permeable, that permits some things to pass and other things not to pass through the cell. So it controls the movement. And this plasma membrane is composed of a very thin layer of a double lipid bilayer composed mainly of phospholipids, but it will also house different proteins that will be different from every organism and every species. And this will form the outer membrane or boundary of every cell. It will control the movement into and out of the cell, and it will participate with the different structure it produces to join different cells to tissues and organs. It will play an important role also to how cells respond to change. Like I said, the fluid lipid is going to be bilayer, it's going to be composed of phospholipids. Its polar end has a phosphate group, and its non-polar end has two fatty acid chains. The polar end tends to look either to outside of the cell or to the inside of the cytoplasm. And the two fatty acids are located, both those layers from each phospholipid will be located in the inside of those two or between those two polar structures. Remember, if something is polar, we call that also hydrophilic, it likes water. If it's non-polar, we call that hydrophobic or lipid-soluble because it does not like water. It may have some carbohydrates, which usually is in the surface area and to maintain specifically recognition, and it may house another type of lipid, which are cholesterol in animal cells, which contributes to fluidity and stability of the cell membrane. So the proteins that are attached to the cell membrane, they may be either peripheral, that means they're located either towards the outside or to the inside of the cell, and that means that if we take them out, they wouldn't kill or eliminate the cell because we still maintain the integrity of the cell membrane. While if we have proteins that pass through the cell once or several times, we call them integral proteins. But what are the functions of these membrane proteins? They may serve as a pathway or a channel for things to pass through the cell. They may serve as a carrier molecule. They may serve as a way to accept different substances, so a docking marker. They may be not only a protein, but they may be membrane enzymes. They may be sites for receptors, so different ligands can bind to it. They can serve as a cell adhesion molecule, so bind to different substances, or they can serve as basically proteins on surface that function to recognize self and for cell-to-cell interaction. So here we have a couple of functions of the lipid bilayer. It forms part of the basic structure of the membrane. It has a hydrophilic interior that serves as a barrier between what's outside and inside the cell. And because it's not static, because the fats tend to move a bit, we talk about this responsibility for fluidity of the membrane. And the small amounts of carbohydrates that are found in the membrane, they may serve as a self-identifying marker, which permits us to identify cells if they're from us or not from us. And different cell types have different markers. And they can also be used as a marker for tissue growth. Here we can see in the picture on the top left, A is just one group of hydrophilic substances, but in the cell, because they're interacting with water on the inside and outside, they form a double lipid bilayer, which you can see in the image below. And as you can see, the image to the right, where you have the red blood cells and the nerve, what they're saying is all cells, regardless of which they are going to be then made of cell membrane. And here we can see how cholesterol can be inside or between the hydrophilic, the phospholipids parts, which gives stability and fluidity to the membrane. And as you can see in the right, we can see how membranes, they may be peripheral if they're only part way through the membrane, either to the outside or inside, but those proteins that go through the membrane are called integral proteins. And here are some of the functions of the proteins in the membrane. So they can be ligands and receptors, they can be channels, they can be doorways, they can be enzymes, they can have different functions that we talked about. One thing that the tissues or the cells do is that they create ways to adhere to other cells. So these adhesion bind groups of cells into tissues and packages into organs. So for us to arrange them and to hold them together, we can do it by different means. Either the cell produces something which is called an extracellular matrix, which is basically a glue, and it's basically made of proteins that interweave into this matrix, which can be collagen, elastin, or fibronectin. We can have cell adhesion molecules in the plasma membrane. So these are molecules that stick to other things, or we can have specialized cell junctions, basically tight junctions, gap junctions, and adhering junctions. Either way, this creates two environments, one looking one side of the cell, and one may look at the other side of the cell. And we can talk about this as the one side looking at the external environment, which can be outside the body, or it can be looking into a tube, like the bronchi, looking at where the airways go through, the small intestine where food goes through. And this we call it the luminal side or the apical side. And the other side, the side that's looking downward, we call that the basolateral membrane, which is basically looking at the connective tissue and the blood towards the other side, okay? So between them, we can have, between neighboring cells, junctions that basically do different functions. Basically tight junctions, adhering or desmosomes, and gap junctions. And we'll talk about each one. Another thing to take into consideration is that when we have two cells, if something passes through the cell, we call this movement transcellular flow. So for example, in this situation, we have something like this, in a second, where things go through the cell, this is transcellular. But if something goes between the cell, this is called paracellular flow. And this is part of the reason we'll talk about in a second, tight junctions. So here we have an example of tight junctions, and we can see tight junctions separate or impermeabilize between cells. So if I have a very tight junction, there's difficulty in passage of things through the paracellular pathway. So things will not go through here, and we'll have different concentration or different charge in one area and different concentration or different charge in the other area. And if we have leaky junctions like we saw beforehand, then things could go through the cell membrane. Then we have another type of junction which is called gap junction. Gap junction is made of the protein connexin, and this basically joins or binds two cells together and permits the passage of small molecules like nucleotides and ions through the cell. And these gap junctions are important for something we'll talk about later in another class, which is called electrical synapse. And finally, we have adherent junctions, which can be either desmosomes, and desmosomes may be made of basically these cadherins or these filaments, or that are going to bind two cells together, or adherent junctions, which are made of actin filaments which again bind two structures together. So these basically held mechanically two cells together. Imagine like a button and a shirt. And then we can see these three here. So we can here desmosomes or adherent junctions. They basically are going to act as mechanical pathways to maintain two cells together. And these are the most abundant in tissues that have movement or stretching or that type of thing. Tight junctions, they firmly bond adjacent cells together. They seal off passage between the two cells, and they're found primarily in sheaths of epithelial tissue, like the skin, like the nephron, like the small intestine, and they prevent undersizable leaks within the epithelial sheath. And finally, the gap junctions are so small connecting tunnels formed by connexin or connexons, and they're abundant in specifically cardiac and smooth muscle, and these tissues that need to be connected electrically to each other. So they permit the passage of some things, small things, but basically we want them to serve as a way to transfer charges or signaling molecules between one cell and the next. And all of this helps us to understand the process of cells being able to communicate with one another. So yes, we talked about direct communication, which are the different junctions, gap junctions, tight junctions, and adherent junctions. But we also have endocrine movement or hormone, where a cell produces a substance, a chemical messenger, a ligand, that will go into the bloodstream until eventually it reaches a target cell for it to have an action. Paracrine is very similar to endocrine. The difference is that the cell, when it secretes a ligand, it stays in the same tissue in proximity to another type of cell to act on it. And finally, autocrine, a cell produces a substance that stays in the same tissue and acts either in the same cell or in the same type of cell. And we'll talk about this much better on next week's class. Now we continue with membrane transport. We know that the cell membrane is selectively permeable. So if a substance may pass through it will pass through it. If not, it will not. So two properties of particles influence whether they can pass through the cell membrane. So the more lipid soluble, the easier it is to pass through the cell membrane. The smaller it is, the more easy it can pass through the cell membrane. An example of this can be seen in the image. This pipette drops a drop of blue dye, and we have basically like a I don't know, let's say like an aquarium, and that's separated by a pure lipid membrane. And this dye does not cross the lipid membrane. So you will see that at the beginning, it will concentrate where it was dropped off on, but sometime later, as the molecules move from more to less, which is the normal type of movement, eventually that side of the aquarium will be completely dyed blue. So it will go from higher to lower concentration. But it cannot pass through the other part of the aquarium. Why? Because the membrane is impermeable to the dye. If we do something to the membrane and make it permeable to the dye, then eventually through this process of movement from more to less, the dye will move eventually throughout the whole area uniformly until eventually it's basically diffused through all parts of the cell. And this creates the membrane transport, which we can separate it like this, when a substance may pass through the cell membrane without the need of any type of membrane transport, with either simple diffusion or osmosis. Those are the two types of movement. But if we need a passageway, a channel, a doorway for it to pass, we may separate them into assisted membrane transport, which may be carrier-mediated or facilitated transport, which in this case I'll put them together. And active transport that can be separated into basically primary and secondary active transport. I prefer this division. So we have passive when I'm going from more to less. So this idea is I'm going from more to less. I do not spend energy, no energy. I don't spend energy. I'm just going from what's easy to what's less. And then we have two types, simple diffusion and facilitated diffusion. The difference between them is just that this one needs a doorway, a channel, while this one can just pass through the cell membrane easily. So that's passive transport. Now if I need energy, I use active transport. In active transport, I go from less to more. So I need energy for this to happen, because if not, it won't be able to happen. I have two types of active transport. Primary active transport, which just means I need energy directly, like the sodium-potassium ATPase pump. to take something from whether it's more to whether it's less. And then we have secondary active transport, where I'm going to need the primary active transport to do this movement. For me to take from less to more, I won't spend energy directly, but I will use the energy that was spent here to do that movement. And from that, we have two mechanisms: co-transport and counter-transport. So we have secondary active transport, which is co-transport, and one is secondary counter-transport. And I will explain this later as we go along.